Welcome everyone to this week's edition of the Extracellular Vesicle Club. We have a very special treat for you today with a new tagging method that we're going to hear about and some of its applications. But first of all, I have something very serious to tell you. And I've taken a cue from Elon Musk and Twitter, which is no longer Twitter. And so we decided to rename the EV Club. And so it's not going to be EV Club anymore. It's going to be Theta, or whatever you want to call it. This is going to be the new symbol for EV Club. It looks a little bit like an EV with some cargo inside, right? I think that's a catalytic, some sort of an enzyme maybe that's being carried by the EV. So if you see an email that's showing up with just Theta in the intro or in the subject line, don't throw it away. It's a valuable missive here from the EP Club. So just a little joke here. And before we start, I do have a couple of announcements for ISEV. So as some of you may know, there is a biomarkers workshop that's going to be held in Denmark in October. And this workshop is open for applications, and you have until Friday of this week to submit your application if you'd like to join that biomarker workshop. It's free for ISEV members, like all ISEV workshops, but you do have to apply for it and be accepted, and then you're responsible for your travel there too. And the other announcement actually has a bit to do with the area of interest of our speaker today, and that is that there is a new special interest group on EVs in nervous systems. So whether you work on the CNS or the PNS, there's going to be a meeting in Rome, a kickoff meeting of this special interest group, and that will be at the beginning of December. So save the date there. You can go to the ISEV website and find out more about that. And the applications for that are going to be opening, I think, at the end of this month, July 31st. All right, so today we're going to hear from Tracy Bale, who's a professor at Colorado, actually at Anschutz in Colorado. And so Tracy is a director there, and has done some very interesting work recently on EVs in reproduction and intergenerational effects of EVs. So we're going to hear about this soon. But Tracy, I understand from your bio that you're also very much engaged in community outreach and getting community involved in our things. So I'm wondering if you would maybe say a few words about that before you get started.
Yeah, sure. So first, thanks so much for the invitation. This is such as we were discussing before everyone got on, what an incredible resource this is, because EVs across so many different fields, and it's advancing so rapidly that this is really a tremendous resource to keep up on all the latest and greatest. So thank you. I was at Penn for 15 years, and I see a couple colleagues on here. Hi, Becky. And then was recruited down to Maryland. And as part of my recruitment was to, as a neuroscientist working in the area of affective disorders and neurodevelopmental disorders, I wanted this center that they asked me to start to really be integrated in the Baltimore community, where there is a lot of trauma and a lot of intergenerational trauma, especially that a lot of individuals, both at Hopkins and Maryland, think about both in their clinical practice and their research. And what I had hoped to do with starting that center was to find ways to integrate into the community. And it was an incredible opportunity to work. We started programs in some of the schools in Baltimore, teaching kids about the brain and bringing in their family members and staff, and then the community. And that led to all kinds of partnerships with a local artist, which if I had time, I would show you some incredible pictures of the mural on some of the schools. And then it sort of amplified from there, where because of something we were doing, it got the attention of the mayor. And so the mayor came and worked with us. And then of course the press comes because the mayor's there, and it sort of just became this snowball of engagement. And I ended up working with city council, which was a whole great but very new experience of working with policymakers and talking about the brain and the effect of trauma. And Baltimore City is the first city in the country to pass a Trauma-Informed Care Act, which means any entity in the city that receives city funds, state funds, or federal funds has to undergo trauma-informed care training, which is super impactful to understand the effects of trauma on the brain. And it really has gone from there to this particular city council member, Zeke Cohen, who then took it to the state level and also passed it at the state level. So really making huge strides and so super exciting to take something and start with kids. And all of a sudden now you're working, at the level of policymakers We also had a trauma summit right before I left, because we were just recruited out here to Colorado, where we brought in PTSD and trauma researchers from across the country and city leadership as well, and held kind of a first of its kind experience of having city leaders talk about trauma, what they do, what the policies are, and then have the scientists talk about the science and learn from each other, which was really a great 2 days. The mayor came, the chief of police, and if you've watched anything about Baltimore, any of the TV series that have come out on some of the violence in Baltimore, you'll know that having the mayor and the chief of police in the same room is pretty unusual. And so it was really impactful. And I think a lot of my colleagues went back to think about the impact of what they're doing in their research, which I think we all should think about.
That's a fantastic message. And thank you for all the work that you did there. We're sorry to have lost you here in Baltimore.
Yeah, no, Baltimore is a great city. If you have the opportunity to visit, it really is a great city.
Yeah, so it's something that I think definitely that we all have to think about as scientists, including EV scientists, to communicate, just like our EVs do between cells. We need to communicate with our communities and work with others. So Tracy, thanks so much for joining today. And I'd like to invite you to share your screen and go ahead with your presentation. And while you're doing that, I just want to remind everybody to please put your comments and questions in the chat box, and we will get to you at the end of the presentation, and we will allow you to unmute yourselves, show your video if you'd like to, and talk with Professor Bale here.
So thank you again so much. To me, this is an exceptional opportunity, because we've been working in this area of EVs across a number of different projects that I'm not really going to talk about today, but for probably the better part of 12, 13 years, and we kind of stumbled into it. For those who work in the reproductive area, you know that Yoel Sadovsky has been doing work in this area with placenta EVs for a long time. And I was at a talk of his years and years ago, probably 14, 15 years ago, when I first like, oh my gosh, what are these things? They're magical, how exciting. And just started thinking about them in the processes of communication I work on stress. So let me back up a minute. I'm a neuroscientist and I work in the area of stress and its effects through epigenetic processes on really brain development is ultimately what we care about. And as our work sort of continued along in looking at the placenta and the effect of maternal stress on placenta signaling, I sort of thought back to what I'd learned from Yoel's presentation, and some of the genes that we had identified really spoke to changes possibly. And that's sort of how we got really into this. But I have to say, in neuroscience and in psychiatry, which I'm now in a psychiatry department, and have several grants now that involve both basic mouse models and humans that cross back and forth between the two in a really translational way, including the project I'm going to tell you a little bit about today, that EVs have really surfaced, I think, in psychiatry as a potential really exciting biomarker. And that's the direction that we're really excited about. For this talk today, because Ken asked me to speak about this paper, this tagging, I'm really hoping this is an opportunity with this audience of experts to talk about, is this a useful, right? This is just a construct, and the validation of it in this paper. We haven't made the mouse yet, and I would love to hear from my colleagues here, like if we think it's worth it, if we think there's a better way to approach it. We've been struggling. I know the field has of ways of tagging EVs and following them, right? And that's so important for studying their importance. So anyway, I hope that that's something that comes out of this conversation today. So as Ken said, I'm at the Anschutz campus. I'm in the Department of Psychiatry now. I'm a stress researcher. So whether you think of stress in rodents, we mostly work in mice. We do a lot of cell culture, as you'll see, as well. And we also do translational work in humans, where we would say trauma as opposed to stress. I'm going to open. I don't want to miss out. If somebody, I don't know, Ken, are you watching the chat? Just in case there's something I need to know about, if someone has a question.
Oh yeah, no, I will definitely be monitoring
Great.
the chat, and you can just go ahead. If anything is urgent, I will let you know. Perfect.
Yeah. I'm happy if anybody has a question, please don't hesitate to interrupt. Okay, so I just want to give you a quick overview of why my lab got into EVs and how we think about them. And I'm sure, like many people on here, are in the last 12, 13 years has really transformed how we think about EVs. I mean, we initially thought about them as, you know, decades and decades ago, of course, as just cellular secreted garbage, and that has changed. But I think in the last decade, it's become remarkable. I think a lot of it owed to cancer biology has done such a tremendous amount of work in this area. We've learned so much from cancer biologists. We as neuroscientists and interested in stress and trauma and its lasting effects, both within a generation and across generations, and how it affects brain development and therefore puts you at risk of neuropsychiatric disease, we first have to identify who's at risk. And so this is kind of a general philosophy of this area of trying to understand neuropsychiatric disease, which is that ultimately it's an intersection of your genes. So those can be different if you are male or female, right? They can come from X and Y chromosomes, which definitely crosses into this area of how EVs and their composition are regulated. Intersecting with your environmental exposures. We're particularly interested in stress or trauma, but that could be anything from dietary effects, whether you're in an urban environment where there's not healthy food source, et cetera, during pregnancy, all sorts of environmental perturbations or enrichment as well, of course. The D is the developmental windows. So when do those experiences and exposures happen at critical times? We know that the preconception environment, which I'm going to talk about, is very important. Pregnancy, obviously gestation, very important time period of development, and then the postpartum period as well. So all of these developmental stages intersecting with the genes that you're at risk, your risk alleles, as well as the environment that you're exposed to. So that's a very complicated model to think about, but it's how we really ultimately think about neuropsychiatric disease. Over the many, many years of research in my lab while I was at Penn and intersecting with so many of my excellent colleagues, again, hi, Becky, and thinking about environmental exposures and in the preconception environment, especially, even as a neuroscientist, you can't sit down to write a grant or think about mechanisms without thinking about what mom and dad bring together at sperm and egg. What kinds of signals do they ultimately bring together that might change, whether they be specific to genes at which that direct the outcome of fertility and implantation and development, or whether they be general signals, which is typically how we now think about this, about the rates of development. I think the rate at which post-fertilization development occurs and implantation are really important ultimately for how the brain develops. So in every grant that I write now I have to, end with the brain, but I have to start way, way back and thinking about these signals, especially. And we all know that there's incredible epidemiological evidence, both for mom and dad in the preconception environment. We actually have grants in both female preconception environment and male preconception environment in both mice and humans. And there's a lot of data going way back in epidemiology, looking at the Dutch hunger winter data, which is now in its third generation. And actually, we have a recent opportunity presented to us to use some of the samples from the Dutch hunger winter, which is, I think, super exciting to think about EVs in those samples. And the Swedish famines, which there are multiple, and the data that collected from there as well, pointing to, yes, in fact, the experiences and exposures across, whether it's stress or, again, nutritional stressors across developmental windows, can have lasting effects. We think largely, especially as it relates to germ cells, at the somatic cell level is where the epigenetic signals are that then ultimately affect signals that are carried by the germ cells. So this is a big field of study. There are incredible researchers, and for this brief talk, I'm not obviously going to go into all the history of this field. This is one of the models that led us to the HA-tag that I'm going to tell you about. So I just wanted to give you a quick summary. Over the last 10, 12 plus years that we've been working on paternal, so male mice stress experience, and you name it, we've done it. We've done different lengths of stress. It's very easy to stress mice for those who don't work in the stress field. Mice are not particularly intelligent creatures. They're very reactive. They're stressed by just about everything. The cologne or perfume you wear, looking at them, picking them up, moving the cage, everything stresses them. We do what's called chronic stress, and we've actually moved sort of away from the daily interaction and coming up with different stressors to actually using a designer receptor model, the DREADS, where we can put these DREAD receptors into stress neurons, but I don't have time to talk about that today. But we've done different lengths of exposure, different levels of exposure. We've done all of that. That's all been published work. And then we've identified changes that happen in development, starting very, very early related to implantation and all the way through looking at how changes in how the male and female brain develop and how they then respond to stress as offspring, as well as going backwards into, again, this level of the sperm and asking, so what's different about the sperm? And then even further back and asking, what are the signals from dad, after dad? This is really important that it's after the stress is over, which I think epidemiologically is very fitting for after a war is over, after a famine is over, is where you see these changes. So there's a lot of cellular work now looking at that, which is defined as allostasis in those somatic cells of dad's reproductive tract. So we've done work going all the way back, trying to look at, can we define these mechanisms of how the cells signal to the sperm and what are the signals ultimately that the sperm carry to fertilization. And EVs obviously is one of the main targets that this field has focused on, ways at which somatic cells in the reproductive tract can offer that information to the germ cell to carry to fertilization. Okay, so this is a schematic that sort of outlines how we think about this now in the field. As you can see on the far left, those are a testis that has the epididymis on top of it there. The very top part is called the caput or the head. These are tubules. This is a cross section in the middle there showing the epididymal epithelial cells that line these tubules. And these epididymal epithelial cells, this point of post-spermatogenic maturation is required, an important maturation stage. The sperm spend a stage in a pH quiescent state there, receiving all of these maturation signals. The thinking in the field now as to how dad's experiences or his cumulative life experiences, both beneficial and detrimental, could be relayed to the sperm that might, if you think sort of over the history of time, evolutionarily might be important signals that they can be carried both by, there's all kinds of growth factors and markers, but there's really a lot of extracellular vesicles secreted into the lumen here. The ways in which the EVs and the different types of EVs intersect with the sperm is really a very dynamic state of study, whether they have a protein-protein interaction sort of a signal. Obviously, the DNA from dad is all wrapped up very tightly in protamines. So is it a protein-protein signal that's carried? Are the EVs actually taken up into the sperm? This is another question. Many think that they are. How do they, as they further transit dad's reproductive tract, taking up more EVs coming from, so this is the caput, but there's also EVs in the cauda and the prostate. As they enter mom's reproductive tract, there's also other EVs. So it's a very complex way of modeling. Like, how do we think about how these EVs and their signals are transmitted? Very complicated. Okay, so that's sort of how we think about intergenerational transmission occurring. I had a brilliant graduate student while I was at Penn, Jennifer Chan, who was working on this project, and she was a pharmacology student, and she spent a lot of time trying to take what we knew in vivo and move it to in vitro, which we defined as called stress in the dish. And the reason we did that is because you can't get a pure population of EVs from the caput epididymis in a mouse. We tried. I actually sent her to a great lab in Canada and Montreal to try and figure out a system. And there just is no way that the tubules collapse. There's just, it's not, you're not able without grinding it up. And we all know there's certain issues with grinding up tissue to collect EVs as well. Anyway, so she developed this, what we called stress in a dish. So these are DC2 mouse epididymal epithelial cells, which we can culture. And Jen spent a few years of her thesis working out the conditions where we take stress hormones, which is in a mouse corticosterone, and mimicking in vitro what we knew to be found in the sperm from paternal stress in vivo. So it was a huge amount of work. Right here is probably 3 years of her life. And I'm not going to spend a ton of time going through all of it, but just to say that she validated this in vitro model with the timing, the concentration, the timing post-treatment, because we knew from our studies that dad doesn't pass on an effect to his offspring if you breed him during stress. It's really in this time post-stress that, and so she was able to mimic that in this culture dish. And this is just showing you the changes in the RNAs, the small non-coding RNAs, and the changes in the proteins of these EVs following this treatment that really nicely mimicked what we saw in the sperm from stress dads. On the left here on this slide is work from that same grad student, Jen Chan, who's now doing a postdoc at Mount Sinai. And so she really whittled it down to these somatic cells, which we call EECs, these DC2 cells, which mimic, of course, the caput of the epididymis in the mouse. We know that it's stress hormone related. There's probably other factors that are important as well. Stress is not just glucocorticoids, but you can't be stressed without glucocorticoids, let's put it that way. So she really went in and defined the role of the glucocorticoid receptor. And so this is a beautiful heat map, which she basically was able to show that if you limited the ability of the glucocorticoid receptor in these epididymal epithelial cells to respond to the stress, you could prevent the passage of the effect to the offspring. And this is just showing you the change using ribotag in these epididymal epithelial cells. You can see a dramatic effect across having stress plus a reduction in GR. On the right is work that an MSTP student who did her undergrad at Penn, but started her work at Maryland, did her first 2 years of medical school and then joined the lab. Nicole Kanyuk, or now married is Nicole Moon. And she's doing her thesis work following up on Jen Chan's work. And this is actually using a cut and run method, which then took this work even further to say, okay, so where is the glucocorticoid receptor acting? What is it modifying? And so this is really getting down in the weeds of these somatic cells. And the reason I'm bringing this up to this audience is because many of you are probably aware that the intergenerational work, the intergenerational field of how information is transmitted between mom or dad and offspring is incredibly contentious. And a lot of that has to do with really a lack of our understanding of the molecular mechanisms, right? So we know that it's very difficult to think about how stress, for instance, could directly affect a germ cell, but this evidence that I'm showing you here is focusing now on the effect of the epigenetic changes in the somatic cells that are important for signal differences to the sperm or the germ cell itself. And so I think this is where the field really has gone. Hopefully it will make the field a little less contentious. But so this is the work that Nicole has been doing, where she identified in that exact same model that Jen developed, the stress in the dish, that there is enormous differences in these cells at this H3K27 trimethylation mark that intersected with the glucocorticoid receptor and ultimately affected mitochondria, but I'm not going to go in that today. Here's the mitochondria data. We don't really have time, but just to say there is a functional readout for these cells, which makes sense. If there's been a bunch of stress in the environment, regardless of the source of the stress, right? The cell doesn't care what kind of stress it is. All it knows is what the hormonal exposure is, and that the stress has ended. And now this is sort of a rebound effect of the cell. And that these changes, especially in mitochondrial respiration, largely then impact cell function and ultimately the secretion of EVs and their composition. So this is kind of the last part I'm going to say about this, which is Jen did the ultimate experiment of causality, which was to collect these EVs. So on the top there, it's the stress in the dish model, the timing, everything the same as we do in vivo, collecting the EVs from the culture medium, of which she has done tremendous characterization of these. So she has vehicle treatment versus the glucocorticoid treatment. She collects the EVs. She then takes takes a male and collects caput sperm to make sure that the caput sperm are matching up with the EVs from that same somatic cell, right? Because those are epididymal epithelial cells from the caput. So the sperm is from the same dad. She divides it into two pools, and the pools are then incubated with the EVs and then washed. She takes a mom, so the same mom eggs, superovulates, harvests the eggs, and then we performed ICSI, intracytoplasmic sperm injection, with these sperms. So she can actually, sperm from the same dad, eggs from the same mom. The only difference is what the EVs were that they were incubated with. And then ICSI is performed, and then these embryos were transferred into either the right or left side of the same uterus. So without now controlling for all other experiences. And then she did two interesting things. She then either sacked the animals mid-gestation, so E12.5, and asked all kinds of questions about, is there any difference in the development, largely pointing to rates of development, as I said at the start of this, or let them grow up and asks about the functional outcome. And do we see the same phenotype in those offspring that we saw with the actual paternal stress? And the answer is, as you can see here, this is all published work from Jen, that in fact, we saw lots of changes in the brain, which is what our main interest was. She also did all kinds of other work at the level of placenta, et cetera. But yes, there's lots of changes. And again, what they're pointing to is the fact that if you change, right, there's been lots of stress in the environment. The stress is now over. And if it was war or famine, the first thing evolutionarily that you'd be concerned with is repopulating your species. And so it would make sense that the signal here would be to speed up the rate of development, which for many tissues may not be any detriment, but for the brain, because it is so unique in its development and that it be exact timing, that may be the effect that we're looking at in some of the phenotype of these animals, that it's not a disease, it's not something that the animals are going to die from, but their brains are significantly different in how they react to their environment. And on the bottom there in E is actually how we assess stress in all mammalian species. This is where we give you an acute stress. I could do it in humans, we could do it in mice. This is a 15-minute restraint, and we just take samples of blood from their tail over that time. And what that graph is showing you is how they're responding to that restraint. And it shows you the exact same phenotype we saw from offspring of paternal stress, which is that they're less stress reactive if they came from cord incubated EVs with their sperm. So this basically is where we left off with, okay, we have a model, we can transmit it across generations. We know that EVs are involved. We've been looking at it. Does it translate to humans? And this is a study actually we did in collaboration while I was at Penn with Neil Epperson, who is now chair of psychiatry here at Colorado. We have now a grant following up on this. And this work was done with Penn medical student and undergrads who were recruited and had never been done before in human sperm, to ask within and between subject, what is the continuity in the small non-coding RNA that we suggest may be coming from both the normal reproductive process of maturation, but also in spermatogenesis, but also be delivered from the EVs, right? You can collect EVs from the semen, the seminal fluid as well. So this was a very complicated study. It took us quite a while just to do the bioinformatics. I would say Chris Morgan worked on this for the better part of 2 years when we were in Maryland, not just isolating the RNAs and sequencing them, but modeling, because what we had done with these human subjects was to look at perceived stress in the prior month. They came in over the course of 6 different collections, so every month for 6 months. And we did it over the course of things like final exam stress, et cetera. But within the psychiatry department, they did a full evaluation every month as to their prior perceived stress. And so then we did this very complicated modeling. And what came out of that was some really interesting data that we're now following up on. This is just showing you a subset of our subjects. So the subject is above in the graph. So 13.2, 22.7, that's just their subject number. And then you can see each row is a different small non-coding RNA, either microRNA, tRNA fragments, or piRNAs. The light blue background is actually sequence. It's not just a blue background if you squint at that. So it's all of those RNAs sequenced in each subject sample over the course of 6 months. So that was great because we could look at the continuity. What are the RNAs that are consistently lowly expressing and don't change much? What are the RNAs that are consistently high expressing and don't change much? And the ones we were interested in for the effects of stress were the ones that transition between low to high or high to low. And then within subject, and then also between subjects, right? So it was a very complicated model. And what Chris determined was a couple of things. One, that it was time +2. So 2 months following the highest level of perceived stress predicted a change in a number of these RNAs, and I'll show you those in a second. The other thing that you might notice, and so those are all the dark blue, sorry, on each graph, the ones that fit the model are in dark blue for each subject across each of the RNAs. But what you might notice, especially in the dark blue that's apparent, is that for microRNA and piRNAs, we have a lot of cyclicity, which is really interesting, that there may be some aspect. And so we're now following up on that. Nicole is now looking at how that might relate to aspects of phenotype of the sperm or in relation to fertility. But these are the RNAs that fit the model. The total number, the dynamic, what we defined as dynamic, as I said, transitioning from high to low or low to high, and then that fit the perceived stress. So we found five in the microRNA several of the tRNAs, and actually none of the piRNAs ended up fitting the model. But you'll notice for those who work in the reproductive field that several of those microRNA we identified were already known. The LET7F and the miR181A already known to be very important for different reproductive aspects. So that was exciting. Like I said, we're now following up on this. We have a grant that has mouse mechanistic studies to examine effects on embryo rates of development, and then replicating the model. As you might imagine, the students we recruited in Penn to do this, as we did a full workup in psychiatry as to their prior life experiences, their adversity, et cetera, they were pretty homogeneous and not a lot of adversity. And so the grant now is replicating the model in a more heterogeneous population out here in Colorado campus, but also to ask about the intersection or the effect of life experience adversity, the cumulative effect, how that might fit in our model as well. So stay tuned for those data that Nicole is collecting right now, again, in collaboration with Neil Epperson's group. So now going back to, okay, so how do we think about this in EVs? So across many of the projects in the lab, and I'm not going to explain EVs to this crowd for obvious reasons, but we, as I think many people in the field, have been very frustrated when we ask about our in vivo models as to how do we follow EVs around from one cell population. So particularly interesting to us was from the placenta. If you have a change because of stress early in pregnancy versus late, or whatever your interests are, how do you follow those EVs being secreted into maternal circulation? Where do they go? And we know there's a lot of effects on maternal homeostasis. We know that we've changed how we think about EVs a lot as homeostatic signals, and they're very dynamic. In pregnancy, they go up 4 or 5-fold. So what are they doing, and how do we follow them? We've tried a number of different things, fluorescently labeling them or injecting them back into animals, and that works somewhat, but it doesn't allow you to ask sort of in the endogenous question of where, if you had a conditional way to say, we know these started off in the placenta, where are they going to, and how do we detect them? Many groups, I know probably people in the audience, have worked on doing things like fluorescent tags. Those are very complicated for reasons of the fact that they're so big that they often affect the secretion of EVs of different types from cells. So we wanted to develop a tag that could be conditional, but that wouldn't affect the secretion. And so I'm going to show you a bunch of the validation. And please feel free if you think there's something else that we should do. I absolutely want to hear it. Went through all of the iterations about what would we tag, first of all, as a protein, in what would be the most ubiquitous, not necessarily the highest expressor. And that itself is a problem I'm sure many of you have faced as well. I think many people have chosen CD63 because it is so ubiquitous, but it is not the highest expressor. And so when you're tagging it, that is sort of a negative aspect to choosing that one that I think is a consequence as well. Anyway, so we went back and forth on all different kinds of tags. We have had a lot of success in our mouse lines using RiboTag that was developed by Stan McKnight's group at UW in Seattle. And so we decided, why not use that tag? Why not use the hemagglutinin, and why not, instead of making it, because it's charged, right? We had to be careful of that. So instead of making it external tagging on the EV, make it internal. Again, recognizing that this makes it a little more difficult to detect. And I will show you data that actually supports that. So we went ahead with the development of this construct. This is based off of, again, the RiboTag development. It's the exact same construct of multiple hemagglutinins inserted into the intronic region of CD63. We had the construct built by Gene Bridges in Germany. We spent years working up all different kinds of ways, because it's quite a large construct, of inserting it and validating it in vitro. And so that's most of the work that I'm going to run through. And then, of course, we wanted to have a biological relevance to this thing. So this is just the construct developed by Gene Bridges. In D there, again, this is all recently published in scientific reports. Chris Morgan did almost all of this work exclusively and really did some brilliant aspects to make sure that we were validating it and that we were confident that it wasn't affecting any aspect of EV secretion or the cargo. So D just shows you in the DC2 cells, the the control versus the C202 is just the name he gave that particular cell line. He had a million different cell lines going, validating the insertion. so as you can see with the HA tag, CD63 is known to be not great in terms of, because it's so highly glycosylated, you get this spread of it on a Western. And so in both the cells and isolated EVs, you nicely see the specificity for the HA there in both of those. And then that's D, and then C is within the cells itself, showing the ICC for the HA tag in those DC2 cells, which you can see both where it is in the cells fits with where you'd expect it. So up to that point, again, Chris had identified that this line, the C202 line, he followed up on many different ones just for publication purposes. He just focused in on the one. And then on the right there is showing you some of the initial validation steps by nanoparticle tracking, that the concentration is not affected, the size is not affected, and the zeta potential is not affected. So there's no differences between control and the HA tagged in these cells. He then moved into more complicated validation using an ExaView, which is the single particle interferometric reflectance imaging sensor technology, which basically says using identification of this chip laid down with the different tetraspanins, in this case, CD9, 63, and 81, that you use that as the complexity of different types of EVs and the co-expression of these tetraspanins, just to demonstrate again that there's no difference across the pull down or the chip expression of these tetraspanins between the control and HA tag. So, okay, so at this point, we were confident that we hadn't affected the cargo generally. The microRNA cargo is in C, and then the PC plot in D for the microRNA, again, showing that so now we're confident that the cargo of the tetraspanins, so the protein composition or the microRNA composition, has not been affected by our HA tag. So we were pretty confident at that point that we weren't affecting it. And then we moved into more complicated identification of taking the EVs isolated from these DC2 cells, and incubating them with sperm, and then looking at different technologies of detection. And so here's where things got really complicated, because remember, this hemagglutinin tag is internal of the EV. Again, because hemagglutinin is charged, you don't want it on the outside, because that will no doubt affect its secretion and how it's interacting at a cell-cell interaction. So Chris figured out that, of course, we needed zinc in the medium, because zinc is so important to mimic the intraluminal environment that's important for how EVs intersect with sperm. Again, you can see with the zinc, I don't know if you can blow that up, but you can basically see with the ICC for that tag that you're getting an abundance of fluorescence in the sperm head and the sperm head and the mid piece here in A. And then B was using immunogold fluorescent, immunogold electron microscopy, a way in which we could detect. And actually we did this, Ken, at in collaboration at Hopkins with the core there, because our core at Maryland was undergoing transition and was not able to help us. So this was actually done in collaboration with Hopkins. On the top there, you can see in the head, unfortunately, it's probably not able to see here unless I blow it up, but there's white arrowheads showing you the single black dots this is after incubation for 2 hours of the EVs with the sperm and then washing. And then if you do a cross section of the mid piece, you can also see that there is some detection there also of this immunogold of the HA tag. So this is just direct incubation of EVs with sperm, the same protocol we've been using for years for doing our ICSI and our artificial insemination and our IVF. So it probably took us almost a year to work out the conditions for this detection of the HA tag to give us confidence that we were seeing specificity in this model. This is now moving a little bit further. So we decided to start asking, okay, this is a lovely tool perhaps, but does it have any biological relevance, especially if we're going to write this up into a paper? So we decided to use IVF. So again, repeating sperm incubation with either control or HA tagged EVs, and then doing the incubation, and then 6 hours later, looking at the transition to single cell embryo. And what I'm showing you there on the top is control versus the HA tag. And again, you can see the specificity with that arrowhead there. You can see it within the single cell embryo. And then on the bottom, Chris included there in that same set of samples with the Immunogold EM that you can also see a sperm that is still present at the intersection of the embryo there post-fertilization. And you can still see the positive detection of the HA-tagged EV. One of the questions across this intergenerational transmission, one we frequently get in papers is, okay, so if EVs are intersecting with sperm and they're carrying a particular signal, how long do the EVs stay around and how long are those signals transmitted? With an HA tag, because it's HA tagged to CD63, right? It's not general for the EV, it's a specific protein. That's a really great question. We don't really understand a lot about this transmission. And so it's interesting to see that there's potentially after fertilization, this is 6 hours later, that we're still seeing the presence of the HA. I will say in later stages, we did continue to incubate these embryos and looked, and we did not continue to see, we didn't detect it. Now, again, in this particular type of model, because you're incubating the EVs, the HA-tagged EVs, CD63 is not highly abundant. And then you're incubating the EVs, which we can't guarantee that all of them are HA-tagged. The amount of signal is not high in this particular type of model. So I can't say whether or not, if we had done something where you had a really abundant signal, would you continue to detect it in later stages of embryo development? I would just say with this model, we did not. So that's about what we have now and where we are in this. This is just a summary of all the things I presented about how we think about EVs and intergenerational transmission, and the fact that we really think the effect of the environmental epigenetic effects are at the somatic cell level, affecting everything down to the level of mitochondrial respiration, which intersects then to affect cell function and secretion and cargo of EVs. And that is likely the signal then, at least at the level of the caput epididymis, to intersect with sperm. How those sperm ultimately carry any type of that signal to the level of fertilization in vivo, we don't know. We did try a few times and then just had to move on because at that point the lab was moving and we couldn't continue to repeat. But we tried doing artificial insemination in vivo and then fleshing out embryos. And that was just a very, very, again, not to say that it wouldn't work, we couldn't have detected it, but we ended up just doing IVF just to move this along and look at the biology. So that's sort of how we think about this in this field now, about how intergenerational transmission involves EVs. Ultimately, what we were able to demonstrate with the CD63 HA tag in these EVs that does seem to have a biological relevance, and that we can continue to detect it at the level of the single cell embryo. There's many different questions, of course, that we could ask. The ultimate one is, in a conditional sort of setup, is there enough evidence here? Do we think it's valuable enough to go ahead and make the mouse? And, you know, in some systems, for instance, the placenta and in pregnancy, you know, where there be a greater abundance probably of EVs that are tagged, could you then, even at a level of Westerns, Ken and I were discussing this early on, you know, grind up mom's liver or skeletal muscle in terms of glucose regulation, perhaps, and detect the tag to say, yes, in fact, we see a transition from the source of where the EVs are coming from to where they're going to that might fit with the phenotype you're seeing in your particular model. So I'm love to hear any input and feedback. We've had a lot of interest in funding from across NIH institutes on EVs as both mechanistic and biomarker potential. So I feel very fortunate for that. A lot of incredible collaborations that have happened across these studies. And Chris Morgan, who now is working pretty high up at the FDA. So he transitioned from academia to a position at the FDA, did all of this work with collaboration with some members of the lab. But so he did all of the work on the tag, the validation, the in vivo, and then he did also all of the work and modeling the human sperm studies as well. So a tremendous shout out to him and to Jen Chan for all of the work that she did initially in developing the stress in the dish model. And then now Nicole Moon in the follow-up and all of the work that she's doing, and an actual paper right now that she has out under review looking at more the mechanistic question about affecting sperm motility and brain development. So Ken, happy to take questions if there are any, or suggestions, or make the mouse, don't make the mouse. Maybe we could do a vote.
Yeah, we could we could even set up a poll here if you want
Yeah,
to.
make the mouse, don't make the mouse. Yeah.
So thanks so much for that, Tracy, for sharing all this work. It's a lot of very interesting things that your lab is doing. So I just want to remind everybody to please put your questions, your comments in the chat box, and then we'll get to you. You should be able now to unmute yourselves if you want to, and to when I call on you to ask the questions. So I see that so Luke has a question here. Luke Belinga, would you please go ahead? I've enabled unmuting now. So Luke is asking,
I can see the chat, Ken. I don't know if that helps.
Yeah.
Do you have results for stress recovery proteins in healthy human sperm? Do you think there are similar results of doing the same approach by proteomics? Yeah, these are great questions. We have so many things now ongoing, and every time I give a talk, I'm also president of the International Brain Research Organization, so I'm frequently traveling around the world, and there's always somebody who has a fascinating cohort of individuals with different exposures, experiences, trauma, adversity, et cetera. Lots of really interesting questions of not just something that might predict adversity, but yes, what about enrichment? What about how do you alter it? So in our mouse model, we have gone out, 3, 4, 5 months post-stress experience, and it never reverses, which has surprised us. And so we've kind of gone with the philosophy in the lab of like, well, if we can really nail down the molecular changes, then we would consider how would we reverse it. And I think we're kind of at this point now, which I've had lots of great conversations with colleagues about, like, what would you consider to be the opposite, right? So if a cell is responding to stress, and let's say glucocorticoids, what's the opposite of that? And, you could do enrichment, but, to mice, I don't know. So we've sort of begun thinking about how would you reverse this signal to then rescue a phenotype. In the human studies, we're really just now at the point of validating the model and then asking about adversity. We haven't really gotten to the healthy differences, although I will say some of the things that Nicole's working on now, looking at just level of sperm phenotypes before we isolate the RNAs and the EVs from the seminal fluid, is sort of suggesting that there is, and again, we're in Colorado, so there's a huge healthy population here, right, of people who are like mountain climbers and big runners and bikers. And so these are some of the questions that we can ask. Interestingly, you might find, because we're in a psychiatry department, we talk about stress and adversity a lot and how we might do sort of acute perturbations, like have subjects come in and look at how they're dynamically responding. Post-pandemic, everybody is so stressed. It has become difficult in this area to find ways to acutely stress people when you bring human subjects in. So there's a lot to think about. I'm a little fearful of testing our model right now because it's post-pandemic. And of course, the study we did on the human subjects before was pre-pandemic, that we may find something that's really just a level of change because so many more people now are on medications, psychotropic medications. So many more people now, I'm in Colorado, there, many things are legalized. And so there's a lot of usage there. So there's lots of things to think about as we move forward in those comparisons in that model.
Yeah, good points there. I mean, the difficulties of controlling, in any human population, but these have even changed over the past.
This has been for replicating our model, which is aim 1 of our grant, involved, in our power analysis, a certain number of subjects to be recruited, but exclusions where because we're studying stress, you can't be on psychotropic medication, you can't be on an antidepressant, et cetera. We have had a really tough time recruitment, not because people haven't signed up, but because they don't qualify. It's really, really eye-opening for the effect of the pandemic has had. Wow.
Let's move next to Rebecca Simmons at UPenn. Please go ahead.
Hey, Becky.
Hey, Tracy. Great talk. So I just wondered if you all had profiled cargo in CD63 compared to your other group? Just thinking about how different sizes, different types, et cetera.
Yeah, that's a really great question. We did not do any kind of an isolation, like a pull down of the CD63 EVs. We've not done that yet, so no. But that's a really interesting question to think about. We don't see a size difference, but that may not be telling because we're looking at the whole population of whether or not the exosomes specifically. We have done, in addition to how ultracentrifugation processes, we've also done size exclusion comparisons. So at a general level, but in terms of cargo, no. Yeah, it's a good question. We have not done that.
Great. Okay, let's go next to Yvonne. Yvonne, you have a couple of questions, so please feel free to ask them both and any others that may come to mind.
I don't, honestly, I could ask. That was an epic talk. Thank you very much. And I have so many questions, but I will limit myself to one of both. So I'm going to preface this by saying I haven't worked in this field for a really long time, but I always found it really interesting. And there were some studies ages ago about methylation status of offspring of stressed, well, not necessarily stressed, but unmedicated depressed mothers. So they altered the methylation status of the glucocorticoid receptors in their offspring. So my question was, do you think something similar happens in terms of egg biology? Or do you think this is, given that the life cycle of an egg versus a sperm are very different, do you think similar things happen?
Yeah, these are really, really great questions. One of the reasons we went into the paternal side of this, 10, 15 years ago was because the complexity of mom, right? Mom, and this great slide that I used to use, this cartoon of like all the factors that you have to consider when looking at mom or egg, of her life experience, but she's also the one doing the gestating, she's the one doing the parturition, all of those factors. Whereas for dad, you can sort of isolate in a more, but then that actually led us to the human study we did, which you really couldn't do easily in a mouse with repeated sampling. So it's one of those examples of like, that was actually a study better done in a human than in a mouse, of looking within subject over time at the changes. So great questions, but when you're dealing with sperm, you get millions of them. And this is something we think about in this intergenerational field. Steve Krawitz and I have had all kinds of debates, like collegial debates, but like, okay, but it's, one sperm. How can one sperm's content possibly be meaningful? Like, yeah, but thousands of sperm show up at the egg. And if there's any sort of EV interaction, wouldn't that be interesting at fertility? But at fertility, there's one egg. And so you start thinking about how would you, because we know a lot about the EV secretion also that happens, coming from mom's side as well. And there's tons of excellent work out there about mom's reproductive tract all the way up into and including the egg. But the egg is just a single cell. It's a huge cell, but it's a single cell. And so it's a little harder to think about those kinds of questions at that level. Not impossible. And there are people way more talented than myself working at ways to get at that single cell question about what is different.
Excellent.
Yeah, it's a great question.
Well, I think the guys have got so much more that they can get rid of on a regular basis. We have a limited supply.
For sure. Yeah.
So my follow-up question is a little bit left field. So in terms of, you mentioned right at the beginning, some studies that I had not heard of, the big starvation study type things?
The Dutch hunger winter or the Swedish famine?
Either or. So my question was basically, do you think that stressors like that are going to produce different effects compared to like psychosocial stressors? And
again, this is a huge question in the field of neuroscience and psychiatry, probably across many different fields. I mean, I know that Becky and I have probably had these conversations many times over the years. Does the type of stress or adversity matter? So Becky's done incredible work throughout her career on everything from maternal dietary challenges, and those could be both directions, right? There's studies that have done things of manipulating protein. There's studies that have done things manipulating fat. And we now know oftentimes in those fat manipulations in rodents is really a lot about fiber changes and short chain fatty, but there's so many. I try to think of it as a stress researcher is that epididymal epithelial cell, does it care? Certainly it cares about the signals it's receiving. And the glucocorticoids, really important, right? And we could talk all day about the fact of your circadian rhythm signal disrupted by stress is all about the glucocorticoid, right? So that's really important too, especially to the mitochondria. But I'm sure Becky would say, like, yeah, there's all kinds of metabolic signals, right? All kinds of signals that the cells see and largely affect. And again, it's probably the level of those signals that change, right? It's always about the delta, but also the longevity, right? You need to have a persistence of, right? Just an acute stress is not going to produce these effects, right? So there's some cellular process that knows when it's been a long or persistent enough change in whatever that signal is that you might be interested in. Becky, do you want to add any piece to that?
No, you summarized it perfectly. It's just like every field. It's really complicated. I do think though that you know, all of the work that you've done, Tracy, with stress is really critically important because, you know, starvation is a stressor and so was COVID. And so those are very different biological types of stressors, but they may converge on similar pathways. It's not going to be the same, but I wouldn't be surprised. I mean, in all of our models and all of your models, we see some of the similar changes.
Exactly. And some of the same systems are affected, but oftentimes in different directions, right? So this one is improving, this one is, right? And so I think a lot about Becky's work when I think about, okay, so if we wanted to reverse this effect at the level of those somatic cells, what sort of things are enriching? Would a enriching diet, but yet if you go too much fat or not enough fiber, then that's also a stressor, right? So it is
the Goldilocks point.
That is exactly, yes, that's exactly a great point. Yeah,
that's brilliant. Thank you so much. I shall yield the floor.
And there's the difficulty also. I mean, my husband and I were just discussing this when we were debating, you know, how we're writing this grant together about stress, is that, you know, for a mice, again, not super intelligent and everything that's different to them is a stressor. But for humans, like if you choose to go on roller coasters, you are making that choice. Your system may see it as a stressor, but you're enjoying that stress, right? So how is that different? I mean, it's acute, it's not chronic. I don't know if you rode the roller coaster every day, but anyway, right? There's also that, right? The human brain is so dramatically different when it comes to stress.
Yeah, there's those risk-taking type people that definitely probably got some kind of different brain chemistry to the rest of us.
Yep, for sure they do.
We could honestly, I could talk to you all day about this, but I shall let other people ask questions.
Well, speaking of talking all day, we're getting to the end of the hour, but we still have a few questions. So Tracy, are you able to stick around for a little bit longer?
Yeah, of course.
Okay, great. Because Shahrokh, you have a lot of good questions here. Do you want to go ahead with those, please? Not sure if Shahrokh is able to unmute.
So have you ever quantified EVs that reach the oocyte upon these? I mean, again, if there's anybody who has a way in how if we made the mouse, so this may be a vote in the column of like, yeah, make the mouse of figuring out if we tried all different ways of approaching this question biologically with artificial insemination, which works beautifully. But it's the flushing out of, and then detecting it by EM, such a limited tag, right? This is the frustrating part of this model, of this tag, is that it's because it's limited in its detection that just makes it much more difficult. Again, if you're going to grind it up, and do a Western, yes. If you want to slice through single cell embryos in vivo, oh, I love it. This is great. Yes. Please tell us.
Here we go. These are all anonymous too. So feel free to.
Yeah. If you think it's ridiculous and it's totally useless, which is what I worry about.
So I see John on the screen here, John Bissler, and John seems to be very interested in this.
Oh, we got to know.
How would you use it?
Yeah.
So it would be interesting to be able to have a tagged EV for a specific cell subpopulation.
Yeah.
So, for example, I work in the kidney, so an aquaporin 2 Cre could drive the marker. And then the question is, what other cell types do those EVs track to? I've not had the same experience you've had. I've gone to study sections who basically have a hard time spelling EV. They're not particularly interested. And so,
Spelled X.
yeah, spelled
Or
X.
Theta.
So how to show that they're trafficking specifically, that would be fabulous. But then after you just got done saying, well, gee whiz, it's actually a low level tag. So it might be an impossible task.
So when we sat down, it was at one of my postdocs and one of my grad students, Jennifer Chan, and we sat in the office, and I had challenged them with, okay, if we're going to make this construct and you both want it for different models, right? One was male, one was the female stressor. So it was like, okay, so go away and come back and let's vote what is going to be our best protein to tag. And neither one of them wanted CD63, but they both ended up agreeing it would be the most useful for all of the models in the lab. So maybe for different models, as you just pointed out, John, that yeah, maybe you don't want to do CD63. Maybe using this approach might be for your particular system and a really abundant EV protein. And again, then you have to choose between also exosome, microvesicle, right? They're going to be more abundant. That's also sort of a
exactly, but in the end, what you've done, at first the work was fabulous. I could gush a long time about how good.
You're sweet, thank you. It's
Lee,
not me, really. Chris Morgan,
I
just
know.
a stellar scientist that did this work, and he was fabulous.
But in the end, you have a way to track this, which is really useful, right? I mean, the specific EVs to a specific location and tissue, and I think that's really fabulous.
If we had any immunofluorescent expert or gurus on here, because one of our questions was, so let's say, John, in your instance, you wanted to know in the kidney, and it's going to go from one cell type to another cell type, and you wanted to section the kidney and to kind of do a co-stain for the marker and the cell type. Because EVs are just notoriously difficult because they're so tiny, and then this tag is inside. And so we really struggled when we ended up having to go to EM. And then when you're working at EM level, you can't just easily move around on a screen and be like, oh yeah, we're in this, right? So I had hoped that by publishing this, some gurus would come out of the woodwork and say, hey, here's a way to do fluorescence instead. And you can, look across the tissue and detect it. But I don't know if anybody on here is a guru that has some ways to do that.
So
we tried a couple different obvious things, and it just wasn't detectable.
Okay. So what did you try?
Oh, I don't know. It's a long, just the kind of the standard,
standard
confocal. So the confocal for sperm took us a long time because sperm are notoriously autofluorescent and they're a pain to work with. But we finally got those. The zinc turned out to be a godsend, trying to mimic how EVs intersect with sperm. But doing the embryos, we didn't have a lot of luck with just standard immunofluorescent. And we did work with some of the experts at Hopkins, Ken, and they also were frustrated. So that was one of my things, like, do we want to make this mouse? Is it going to be useful to people?
Mm-hmm.
I'm happy to make it.
Yeah, because I think if you see, if you are, if you know the level of sensitivity that you have, that's a difficult question to, that's a difficult,
it's hard to know without making the mouse to say like, okay, yep, we can detect it's, tons of it in the placenta. Maybe the only way we'll find out is just to make the mouse. And if you can take down all the names of the people that said yes.
Yeah, well, you have 15 volunteers here who are going to contribute financially and with hands. So
the money I got.
Okay,
that I'm not worried about. The money I got. It's that I don't want to make the tool if it's not going to be useful to people.
Yeah, exactly. Yeah.
So if the person who said no feels comfortable saying why, I'd love to hear it. Unless if it's just the obvious reason of the detection is difficult. If there's any other reason, is anyone?
What do people not like about this approach?
Yeah.
Anybody?
Don't be shy.
All right. Well, we're apparently shy today, but
I think it's a great approach, Tracy. This is Becky. I'd like to compare it to what you and I know have talked about this with our CD9 GFP mouse. So we struggled also to try to localize this, and we haven't, we haven't found the greatest approach, but we finally have talked to Nicholas Plunkta who's at Penn, when he's in town and not in Spain. And I think he feels confident that he can help us with this.
Okay.
There's also another person at Pen/Chop. And so if this works for us, I'll call you.
Okay. Well, it would be nice to compare.
We could compare because this is
trees.
sort of my question is, we've been really interested in looking at small versus, larger EVs and we've not published any of this, but, and other people have shown that there's really different protein profile. We find very different profiles for RNAs as well. And so and looking at different tags, are those marking specific populations? I mean, there's so much overlap, it's hard to know. And it would be great to see where yours are going, and is this going to be a generalizable tag? Are this the same way? So, because we want to see if they're coming from the adipocyte. So we have an adipoQ mouse that we've created that we've crossed with the CD9-GFP, which we've crossed with a mito tag. So with the idea that we can see if those EVs with mitochondrial cargo coming from adipocytes are getting into the embryo. Me and obesity, I care all about that. So
no, Becky, this CD9. Sorry,
yeah. Go ahead, Ken.
Oh, I just wanted to ask, the CD9-GFP, is that an internal GFP, or is that in one of the external loops?
I think it's in the external loop. I'm not positive about that.
Yeah, it is one of the things that has really been, I feel like, a hurdle for the field of identifying
absolutely
the best ways to do this. And so maybe doing it from multiple ways and then comparing
multiple ways. And then I think it's really the microscopy. So, not many of us can afford those really expensive microscopes, but we'll see if we can detect it. Penn, we wrote this instrument grant to get this incredibly fancy, incredibly expensive microscope. So we trialed it, and we were actually able to see, and we didn't use, I can't remember now if it was our GFP. I don't think it was our CD9-GFP. I think it was fluorescent labels, but we saw down to the EV. It was amazing what we could see in that microscope, but we didn't get the grant, and Penn didn't decide to give us.
So one of the things we wanted to do in this development, and you'll note clearly we didn't do it, which was, I wanted it to be an accessible, usable, and tool that was high throughput. That was my, and so I don't think, although again, if your question just is, going back to John's question about cell types in the kidney, if you had a way to sort those cell types and could run them on a Western, the HA tag would be brilliant. It would work just fine. It's the level of, I want to section through or look at the whole mouse and see where it went. I don't think this tag would, I don't think it's going to be easy to do that unless somebody has a way to penetrate.
Could you visualize? And this is something we've grappled with because we want to ask that question, but in the embryo, looking at, where in the embryo, in the pre-implantation embryo, we thought about doing single cell RNA-seq to look at that specifically.
Yeah. So we looked at this, Becky.
Did you try that? Did that work or not?
No. Well, we didn't do it because after sitting down and calculating how many cells you need, it would have been impossible. We did the calculation of how many mice would we have to do to do this.
Okay. Well,
we talked to Adrian.
But if we thought about it instead of the embryo, and we thought about, okay, let's see if it goes to the liver or to the brain.
If you can grind up the tissue and just want to say, does it go there? This tag would work great.
Well, if you did single cell, then you might be able to. You might. I don't know. We thought about it. We haven't tried it yet to see it, because I think the level of detection and the amount that's getting in is going to be so small that it's probably impossible. It's a pie in the sky idea.
Like I said, that paper, the work that Chris put into the detection and validation was, it was years.
Yeah.
So I don't want to ignore there was a couple other questions. Do you think there might be a difference in X sperm versus Y sperm in terms of, yeah, that is a great question. I love questions related to sex differences, especially sex chromosome. It is not something we have done. I guess we could sort sperm based on weight for X and Y and then incubate. And that's a really interesting question. We have not done that. That's a really interesting question. Mouse with the including HA-tagged request stocks work. I may have missed it. Where are you going? Yeah. Okay. So I think we already covered that. EVs and seminal fluid. Yeah, we're doing all of the seminal fluid stuff now. I will say, when we did that initial study at Penn with the human sperm, we met with the fertility clinic multiple times to say, please don't wash the sperm. Please don't wash the sperm. Please don't wash the sperm. Here's what we're going to do. Here's why. And then ultimately, when they gathered all of our samples, they washed all of our sperm. And so we weren't able in that huge study to look at the seminal fluid EVs. It's what we're doing now. And we have a whole different, much more complex approach to it. So we should get really interesting data on the EVs, correlation with the sperm. We did some initial studies at Penn that did show like the change in EVs in the seminal fluid mimicked the changes we saw in some of the RNAs in the sperm, but we actually are now powered to do that here. So we'll see if that works. Prostate EVs, big question. I've often wondered, right? So this idea that EVs are secreted all along the reproductive tract in males and females. So they can't all be taken up, right? Because then the sperm head would just get bigger, right? So there must be some really interesting way of protein-protein interactions to carry the signals. And then some sperm, we tried doing some EM to see if we could see differences sticking to the, great question. Don't know, lots to ask. I have all kinds of visions of making a 3D modeled reproductive tract and having EVs secreted with different fluorescent colored tags to intersect and see what shows up at the egg. Haven't done it yet, but yes, great questions. Have you tried? No, we have not. And I am writing this down. Glyoxal fixation. No, we have not tried that. We tried a couple different fixation approaches. We have not tried that.
Yeah, I think the super resolution microscopy could really be a breakthrough here because of the single or near single molecule resolution that so that might be something interesting to try out with the tissue.
Okay.
Of course you need the mouse.
Yeah, right. I mean, but nowadays making a mouse is less of an ordeal than, although I will say, unfortunately, this construct that Gene Bridges made was kind of old schoolish in that it's huge. You can't do CRISPR with it. We tried with a group at Maryland who's CRISPR experts and like, oh my God, this thing is enormous. So we did an old school transfection for the paper. So I think we want to sort of maybe change the construct itself, which kind of says like, we don't have to do CD63, maybe we want to try a couple different proteins. If anybody's still on here who has a better protein choice, shoot it in the chat. We might think about maybe, again, we chose CD63 because it was the most ubiquitous, but it is not the most abundant, and that can be a problem. I don't know if anybody else has any other protein thoughts that.
Yeah, but Tracy is asking, what do you think would be a better marker for you?
Yeah. Okay, flotillin. Again, I wanted it to be, I was like, if we're going to make this, let's make it such a useful tool that people in the field across different areas could utilize it. And so that's why we settled on CD63. So. All right.
I guess we'll conclude things here. But of course, feel free to reach out to Tracy with any.
I'm taking a screenshot of the poll so I want to say like, was there someone secretly from my lab being like, no, we don't need this mouse?
Good.
Right, thanks, everybody.
Thank you all.
If you have any other thoughts, shoot me an email. I always love to hear them. Thank you so much.
That's what it's all about. Okay, take care, everybody. Bye now.